AP Bio students. This is Shelton here, just giving you your Screencastify for Chapter 6, your Introduction to Metabolism. So we're going to just go over all the notes. I'm just going to do it really briefly since I'm going over them on our Zoom meetings. But you have this as a backup plan too, in case you want to slow me down and go back through some of the concepts that you should be pulling from Chapter 6. Now remember, before you do take the test, go back through the You Must Knows and make sure that you know what you must know before you take the test on Monday. So anyway, for today, as we go through the notes, we'll go through all 5 sections in order, even though we went through just the first 2 sections in class. I told you we'd go over 6.3 later. 6.4 we went over, that's what your lab is also going to be addressing, more of 6.4, and then we'll also go over 6.5, control of metabolism. So from the presentation, let's take a look here. First and foremost, what we need to make sure that we are understanding is we are understanding what we mean by metabolism. And so with metabolism, what we're really talking about is energy exchange in living things. Now, that energy exchange can be an energy release, that can be an energy usage. So what we're seeing here in this bioluminescence here in the water is living things are releasing energy in the form of light, so in photons. But if we were talking about something like the release of photons from a star, well, that wouldn't be metabolism. It would still be energy. But when we specifically are talking about metabolism, we're talking about these biochemical pathways that are using energy or releasing energy in living things. And that's what all life and all living things have to have metabolism. And those metabolic activities actually get classified as either being catabolic activities or anabolic activities. And essentially what's happening here in a catabolic activity is we're starting with something large, a large molecule, and these large molecules are stored potential energy, and releasing those into smaller molecules, something like sugar releasing into carbon dioxide and water, but then taking the energy out of it. Those are our catabolic activities. But we also need to be building things, and in building things, we're taking smaller units, we're building them into larger units. In order to do this, it's an energy requirement. Actually, when we define what metabolism is, oftentimes definitions are the sum total of all catabolic and anabolic activities in a living thing. Now, other terms that we're going to have to make sure that we're addressing too is understanding that this is an exergonic reaction. Exergonic means it's giving off energy. We'd also say that this is a spontaneous reaction, the fact that the breaking down of molecules naturally occurs. This, on the other hand, would be an endergonic reaction. We have to put energy into it. This is a non-spontaneous activity. And then one other term we're going to learn in this unit too is that this actually has a negative delta G, a negative change in free energy that's now given off and able to use by something that requires a positive delta G that now we have to put into it. So those are some other terms we're going to be addressing in this chapter. Now, we do need to have some basic knowledge of energy. And when we do talk about energy, we do need to recognize when we talk about potential energy, like this diver up here on this platform, he has the potential to actually turn into kinetic energy, this energy in motion or the energy being currently used. Now, when we say energy, energy has the ability to do work or cause change. That's what energy actually is. And energy comes in lots of different forms. But right now, what we need to recognize is potential. Got the potential to release its energy. And actually in living things, that potential energy comes in the form of molecules from chemicals. It's a chemical stored energy. Sugar is a stored potential energy. It's not actually releasing the energy until we start breaking it down, breaking those bonds and getting that energy out of it to do work with it. ATP is not actually energy. We have to hydrolyze ATP in order to then get work out of that ATP. But remember too, with energy, well, we've got to input energy to get to potential, and then we release that energy, and then we've got to input more and its potential energy, and we release it. And so it's a series of exergonic, endergonic reactions that end up happening in living things. Now, when we talk about energy transfer, that's what the study of thermodynamics is. It's a study of energy transfer and energy change. Now, we do need to know the first law of thermodynamics versus the second law of thermodynamics. And the first law of thermodynamics is essentially law of conservation of energy. It means that energy can't be created or destroyed. All the energy in the universe is all the energy in the universe we're ever going to have. It just changes form. And so we take the chemical energy that's here in this fish. Well, it got that energy from somewhere, but we're going to convert it in living things. And in converting it, we'll use that. But that energy is then going to flow through and get lost from the living systems in the form of heat. Molecules get broken down too, but energy can't be created or destroyed. That is my first law of thermodynamics. But we do have to study where that energy goes, that transfer of energy. And where it goes actually is a lot what the second law of thermodynamics tells us. Second law of thermodynamics is entropy. Entropy is randomness or disorder. And what second law of thermodynamics tells us is that the universe tends towards disorder. So we can go from highly organized energy. Well, that energy is naturally going to move towards its more disordered form of heat. Molecules can be highly ordered and organized, but the universe is going to tend to break them down into its smallest, most disorganized form. And so that's randomness or disorder is what we refer to as entropy, and that's what our second law of thermodynamics is describing for us. Which also brings us to this question, kind of an interesting question when we talk about the highly ordered, highly organized living systems. Living things are highly ordered, highly organized, which tends to defy the second law of thermodynamics until you actually start thinking of it in terms of, in order to stay ordered, how much disorder are these living things actually creating in the universe? They're creating way more disorder in the universe as they're breaking things down and utilizing the energy than they are themselves. So if you think in your lifetime, all of the food that you consume, all those organized pieces of matter that you have put in, broken down as energy, just so you can stay highly ordered, you create more chaos and more disorder in the universe than you are ordered yourself. I kind of associate it kind of like with my desk. The more knowledge I order into my brain, the more disordered my desk becomes, is what ends up happening. So gotta keep that in mind. Now, in section two is going to be talking about the concept of free energy or Gibbs free energy. So Dr. Gibbs, who studied this is why he has the unit after himself, capital G, for Gibbs free energy. But what free energy essentially is saying is the energy that's available that can be used to do work. So this is what we mean by free energy. Now, this equation here, this will be given to you on an AP equation sheet, but it's never asked. What's most important, though, is you understand that when we are talking about free energy, we need to know that if it's a negative delta G, this change, the final amount of energy minus the initial amount of energy, when there's that change, that's a negative delta G, this is what is an exergonic reaction. It gave off energy to do work. We also classify these as spontaneous reactions with a negative delta G that's exergonic. These would also be our catabolic reactions, the breaking down that's actually happening. If it has a positive delta G means that we had to input energy into it. It was endergonic. These are not spontaneous. These anabolic activities require an input of energy in order to take place. So if we talk about these, making sure that we're understanding this tendency towards disorder, and also then that everything tends to this lowest energy state is where the universe tends towards. This is the entropy, towards randomness, lowest energy state. This is what diffusion is even based on too. Molecules move to their lower energy state, the more disordered, higher entropy is what that is referring to. In terms of a graphical analysis of these terms, this would be showing you an exergonic reaction where the reactants versus the products, the amount of energy that is contained within the reactants, which is high, the release of that energy. Now my products have a very low amount of energy. Well, final minus initial, I'm going to have a negative delta G in calculating this. So negative delta G, exergonic reaction, spontaneous, catabolic. Opposite of this, so this could be actually up here, if I'm looking at exergonic reactions, we could apply this to aerobic respiration, where we're starting with sugar as our initial reactant, and then through all the steps of aerobic respiration, we get to carbon dioxide and water. That's all negative delta G. Now, if we talk about something like photosynthesis, carbon dioxide and water, in order to convert that into now something larger, like G3P or our glucose molecules, well, I've got to have an energy input. These are endergonic, they require energy, they're non-spontaneous. These are our anabolic processes, positive delta G. Now, one of the molecules you absolutely positively need to know and need to make sure that we are understanding is ATP. So remember that ATP is essentially, it is an RNA nucleotide. So we've got this nitrogenous base adenine, I've got my pentose sugar of ribose, I have my phosphate, but now what I actually have is actually a triphosphate. So 3 phosphates that are bonded together. Well, remember these phosphate groups each have a 2 negative charge to them. So to force a covalent bond between negatively charged ions requires a tremendous amount of energy. So in these bonds here, if it took a lot of energy in, remember first law of thermodynamics, if I had to put a lot of energy to make these bonds, that means when I break the bonds, I release this equivalent amount of energy in that process. Now, in order to go from ATP to utilize this energy, well, this is a hydrolysis reaction. So anytime they reference the hydrolysis of ATP, put water into it in order to now break off one of the phosphates. It's inorganic phosphate because there is no carbon base to it anymore. So this inorganic phosphate, and now I have ADP. But now I have energy. This is a negative delta G. I now have that negative delta G, that energy that's available to now do work in the cell. And the cell can actually couple that with another reaction. So with something that is endergonic that actually requires energy can now be coupled with that exergonic reaction that was produced in the hydrolysis of ATP. So this is also what we mean by energy coupling. So what I like to describe as energy coupling is in the adult world, you have to make money before you spend money. Well, same thing happens in the chemical world. You have to release the energy before now you can use the energy. And oftentimes that release of energy comes in the form of hydrolysis of ATP. If I now couple this formation of glutamine that required energy with the hydrolysis of ATP that released energy, well, now I have enough energy in order to perform this anabolic activity, that non-spontaneous reaction. So that's what we mean by the coupled reaction. Oftentimes in this coupling, what is actually produced is actually the action of ATP is we actually create this phosphorylated intermediate. The phosphate actually gets transferred to one of their reactants, and this makes it a highly reactive. It's kind of, I like to think of these phosphates as kind of like hot potatoes. If I actually, the action of the enzyme involved in this is to transfer the phosphate from ATP onto this phosphorylated intermediate. This intermediate now has this hot potato attached to it, and now that energy forces it to react now with here this amino group in order to now form another molecule. So sometimes what's not pictured here is the enzyme that would also be involved in phosphorylating this intermediate. But this is what's referred to as that phosphorylated intermediate that has this phosphate group temporarily added to it in order to initiate and provide the energy needed for this endergonic reaction. So that's kind of showing you the actual action that's there. Now, ATP gets used for lots of different types of work. I mean, so we talked about active transport. ATP is used for that type of transport, for motor work. And then you also saw it then as kind of that chemical enzymatic work as well. With ATP, ATP ends up just being this cycle where ATP, adenosine triphosphate, through hydrolysis, then we release that energy. So in that exergonic reaction that can now be used for endergonic coupling, now we have our ADP and inorganic phosphate that now we can go back. This is going to be through aerobic respiration where we can regenerate, put energy in, transfer it from the sugars, and transfer that energy back into ATP. And so I think of ATP as a charged battery. ADP is then an uncharged battery. And so we just continuously recharge them, store the energy, release the energy. Oh, now it needs a new charge. And so it just becomes this cycle inside cells of energy transfer. So the big part of chapter 6 that we need to make sure that we are understanding is we are understanding enzymes. Now, enzymes are biological catalysts. So do not just repeat that. You need to make sure that you're understanding what you mean by the definition of a biological catalyst. First of all, enzyme is an organic molecule. It's protein. These are proteins. Proteins have primary, secondary, tertiary, quaternary structures to them. So we know lots about proteins at this point in time. And it's a catalyst, and a catalyst means that it speeds up a chemical reaction. It's going to speed up this change from a reactant to a product. But it's not a reactant, nor is it a product. It's not directly involved in the chemical equation. You're not going to see it in the equation. But what it does is it speeds up this rate of reaction. So it is a protein that speeds up the rate of a chemical reaction. Now, what we need to make sure that we're understanding is how it does that. So how does an enzyme, and enzymes are specific. So enzymes are very specific to each of the reactants that they catalyze. So first of all, before we understand, I'm going to just show you really quickly, essentially what enzymes do is they speed up the rate of a reaction by lowering the activation energy required. Now, activation energy is the term that we need to make sure that we understand first. When I have reactants, so even this, this is a spontaneous reaction that's going to have a negative delta G, just like a boulder that's at the top of a hill. That boulder will eventually come down the hill. But in order to get that reaction to occur, I have to input a little bit of energy. That energy input to get it to go is the activation energy. So just like if I had a gallon of gasoline is potential energy. It has a lot of stored energy in it, but it's not just going to spontaneously combust. You got to put a little bit of energy into it first in order to now get it to go to the water and carbon dioxide and release all the energy that was stored in there. This activation energy that you put into it first before it reacts, well, it's going to actually change those reactants into this transition state. This kind of unstable state. They've absorbed enough energy where they're unstable, where now they definitely want to react in order to become more stable and then release the energy that not only did they absorb, but the energy that they were also storing in them too. When we calculate delta G, we are only interested in the amount of energy difference between the products and the reactants is all we're looking at. This was just the amount of energy required to get the reaction to go. This is not calculated as part of your delta G, which also means too that since enzymes reduce the amount of activation energy required, they do not affect the delta G of that reaction, which is what we should make sure that we are understanding. Now with enzymes, first of all, make sure we're understanding the term. This is showing you a model of a protein. This is a substrate. Essentially, the term substrate just means reactant. It's the reactant that this enzyme is going to interact with. So how an enzyme interacts with its substrate, with its reactant, is every enzyme has a specific active site. An active site is a shape that is essentially equal and opposite to that of the substrate. That is in which it's going to affect the chemical change. Now, we have to also remember that enzymes and substrates are in solution. So enzymes and substrates in solution are moving around in solution and collisions just happen and they're moving rapidly. And so with this and moving very, very rapidly, substrates and active sites will collide. Now with this, and the more substrate you have with enzyme, the more collisions are going to happen. Fewer enzymes, well then less substrate, less collisions. You can just imagine if you were playing blindfold tag, where the person who was it and the ones who are running, everyone's blind and no one can hear. Well, you can imagine what that collision is going to be like. The faster you run, the faster the collision rates. That's how temperature also affects enzyme activity too. But when the substrate hits the active site, it's actually going to temporarily form what's referred to as an enzyme substrate complex, where they actually temporarily connect briefly. Well, in this region in here, in the active site, you've got to remember that there's R groups that are in here too. And those R groups and this substrate jamming into it, it actually creates this induced fit that actually reduces then the activation energy required to cause change in the substrate. So it is actually through this action of induced fit that activation energy gets lowered. Now, enzymes themselves are not used up in the reaction. So substrates connect at the active site. So in this picture here, this would be my induced fit. My substrates are bonding, forming this enzyme substrate complex. this induced fit then initiates that chemical change that occurs, and that also now changes the shape of the protein enough to release the products. The products now are released into solution. The enzyme goes back to its original conformation and now collides with new substrate, and enzymes just get reused over and over and over again. But along those lines, too, when we start thinking about enzymes as proteins, well, this is where we can also address and understand how pH and temperature can affect enzyme action. So, one, when we know temperature, and different enzymes have different optimal temperatures. But what we should know about temperature is we know that temperature is average kinetic energy. So, if I increase temperature, that speeds up the rate of collision. So, sometimes that increases the rate, and we can actually see this with the slope. We can see this increasing rate. But then if temperatures get too high, it can denature that protein, changing the shape. So now the active site no longer functions. pH does the same thing. Remember that with pH, we start changing that hydrogen bond interaction. We start changing the R group interactions too. So every enzyme ends up having this optimal pH where it's going to have, where the protein will have its optimal shape and therefore its active site functions. So imagine with both of these, imagine your key to your house melts. And so now the shape of your key no longer functions. Well, that's what happens when you get out of the optimal pH range or you denature the protein with too high of temperatures. So make sure you're just aware of those. Now, other things that affect enzyme activity, though, is, just think about it this way, is the amount of enzyme you have. And if I have very few enzyme but a ton of substrate, or very little enzyme, very little substrate, or a whole, those concentrations are definitely going to be affecting how quickly in these random collisions we are going to actually get products being formed. So that's what your lab is going to be addressing too, is substrate concentrations or enzyme concentrations and that effect. Now, enzymes being proteins too, remember that some proteins, they do require cofactors or they require coenzymes in order to maintain their shape. So when we talk about like a mineral that needs to be in here in order to form a base, that would be a cofactor. But sometimes these enzymes need another organic molecule, like a vitamin or something like that, to attach to it to have its right shape. That would be a coenzyme, is what that would be. So coenzymes, cofactors, actually vitamins and minerals, and some essential reasons why we need to have vitamins and minerals in our bodies in order to be able to join with the enzyme in order to make sure that it's the proper shape. Now, we can also control enzyme activity. And so in controlling enzyme activity, there are certain substances that act as inhibitors. They stop the enzyme from working. And so actually some medications that we take are actually enzyme inhibitors is what they do. And if you stop one enzyme in one biological pathway, you stop the entire pathway. So inhibitors can first of all get classified as being a competitive inhibitor or a non-competitive inhibitor. A competitive inhibitor is essentially, it is a molecule that binds to the active site and just interferes with this substrate being able to collide and have an induced fit in here. You can also have a molecule that's a non-competitive inhibitor. I kind of call it a backstabber. So it actually binds to a different region, not at the active site. And so this also changes now the shape, kind of like if you think of you got stabbed in the back, you get your change in shape. And so the active site is no longer then the shape that matches the substrate, is what happens with a non-competitive inhibitor. We also have enzymes that are controlled allosterically. And so this would be your quaternary structure here, but here what we're seeing is this is a type of enzyme that has 4 different active sites, and we can have molecules that are either activators or inhibitors in allosteric enzyme activities, kind of like on-off switches. So I can have the lights in the classroom. I've got 12 lights, but all I need is one switch. And with one little switch here, I can activate all of those lights or deactivate all of those sites too with an activator or inhibitor. To activate means to turn the active sites to the right conformation. To inhibit means I'm now shutting all of the active sites, changing their shape so they will not bind with the substrate. But these are just different ways that cells can regulate a type of allosteric enzyme that has multiple active sites. The last thing that we want to talk about is actually, and it's really important that we make sure that we're understanding it, is feedback inhibition. So here is a model of a biochemical pathway. This is a biochemical pathway showing you the enzyme that is producing this end product of isoleucine, this amino acid. And they're showing you that there are 5 enzymes and intermediate molecules along the lines that are required in these steps to make this end product of isoleucine. Well, in this feedback inhibition, I mean, if you're a cell and you've now made plenty of isoleucine, well, you want to stop the production of isoleucine. You don't want to just keep on wasting energy making more and more of this product that you don't need. So what ends up happening in cells is this end product often acts as an inhibitor. And in this case, this is a non-competitive inhibitor that actually binds to the enzyme that would actually initiate these steps, the very first one. So now high concentrations of this end product actually acts to inhibit the first enzyme. So if I stop the first enzyme, that means I don't get any of the rest of the steps happening. But what ends up happening too is if isoleucine becomes low concentration in the cell, well, we know diffusion, molecules move from higher concentrations to lower concentrations. So isoleucine is actually going to fall off of this enzyme when concentrations become low. And now this enzyme is active again, and we can continue with this process. So in feedback inhibition, this is a negative feedback system, but the end product is often what's regulating or shutting down. Excess of the end product is shutting down the entire biochemical pathway, usually acting as an inhibitor to one of the enzymes in the process. So again, with this one, here's just a biochemical pathway. And in feedback inhibition, typically what would happen is excess product D would end up binding to enzyme number 1 and then stopping this reaction from happening at all. And that would be a feedback inhibition. But now if this product D becomes low in concentration in the cell, well, then D would come off the enzyme. Enzyme 1 would start being active again, and this biochemical pathway would occur one more time. Again, enzymes are specific. Most enzyme names, just so you know, end in ASE, so that kind of helps us. And usually the substrate itself is actually part of the prefix, and then the suffix then being ASE. So sucrase is actually the enzyme that is specific to break down sucrose. And so this is the enzyme that lowers the activation energy required to break this glycosidic linkage between glucose and fructose. So that's sucrose. But if somebody is lactose intolerant, lactose, well, you need the enzyme lactase in order to break that disaccharide in order to be able to now produce those 2 monosaccharides. Lastly, before we leave, remember, enzymes are biological catalysts. They speed up the rate of chemical reactions in living things by reducing the amount of activation energy required for that reaction. So that is what we need to make sure that we are understanding, and then make sure that you are understanding delta G versus positive delta G versus negative delta G, etc. So with that being said, make sure you do read chapter 6, all of it. I need to make sure that I'm getting myself out of here somehow, some way. And hold on. And with that, I am stopping this, and I will see you guys back in class.